It is my great pleasure to introduce Professor Staci Bilbo from Psychology and Neuroscience here at Duke. Staci began her faculty career here at Duke, briefly went to a place up north that she didn't like as much as being at Duke, and came back. And we're really glad to have her back. It's been a few years since she's been back. She has a thriving flock of chickens. And more from a research.
You meant the grad students for a minute.
A thriving flock of graduate students as well. So I think Staci's research is just super cool. It is working at the intersection between the immune system and the brain. And if you weren't previously interested in that question a couple of years ago, hopefully you were really interested in that question now. And Staci is really one of the world experts on this topic. She has been working on all kinds of really interesting angles, including the effects of infection on the brain, the effects of pollution on the brain, and all kinds of things, many of which she will probably tell us about today.
Hey. All right. Okay, is this working? Can you hear me? Sort of? Yes, okay, awesome. Well, thank you, Jenny. I'm very impressed with your spur of the moment introduction. And this is really a pleasure to be here in person. I've lectured in person for a while, but I think this may be my first talk in person for a couple of years. So thank you very much. So as Jenny mentioned, I think a lot about how the immune system and the brain talk to each other and how this is important in both health and disease. And my lab is particularly interested in a cell type within the brain called microglia, which are affectionately known as the resident immune cells of the brain. They are essentially a sort of white blood cell called a macrophage that come into the brain during early development and are then the host defense cells of the brain. But beyond this, we know, based on lots of research in the past couple of decades using more modern microscopy techniques, that they're also incredibly dynamic cells. So they sort of wear two hats. They're immune cells, but they're also brain cells, and they're constantly moving around and listening to synaptic and to neural activity within the brain, which brings up many interesting questions about what it is that they are doing. And this is just in a healthy, sort of steady state within the brain. So as I mentioned, they are a type of macrophage, and just like a macrophage in the periphery is really good at eating things, in this case it's about to eat some bacteria, microglia are also very good at eating things. So they have a really critical role, especially in early brain development, in removing apoptotic cells and debris as the brain is building. But one of the functions that has been defined in recent years that has generated a lot of interest in their function is in synaptic pruning. So as you know, you have a lot of synaptic connections happening early in development, and then these are removed through activity and experience-dependent mechanisms. And microglia are one of the major cell types to go in and remove synapses that are no longer needed. So why? One of the questions I've had for many, many years when this research emerged was, why on earth do we need immune cells in our brains to do something like prune synapses, when presumably the brain has mechanisms in which it can do this just fine? And that's true. Well, as we know, nothing in biology makes sense except in light of evolution. And I don't need to tell anyone in this audience that we are constantly plagued by things that want to kill us. So I did have to update this recently to include our new companion. But the fact remains that this is not new. As long as we have existed on this planet, we have had to have mechanisms by which we evade things like infections that try to make us sick. So the immune system is really amazing. It's an amazing, complicated system. This is the least complicated slide I could find for the immune system, which I love. It sort of has all the components there, but actually it's missing one really important thing, which is that immune defense includes behavior. So again, I'm sure everyone in this audience can identify with this right now, unfortunately. When you're sick, you feel like crap, right? You exhibit a lot of really stereotyped changes in your behavior, including changes in cognition, social withdrawal, anhedonia often. The surprising thing is that these things aren't caused by the pathogen itself. They're caused by your own immune system, collectively known as sickness behavior. So again, why on earth would this be happening? Well, we know based on lots of research now that these sorts of behavioral changes are adaptive, at least in the short term, because they help us to recover from infection more quickly. And this change in social behavior in particular is of great interest for my lab because we know that many neurological disorders, neuropsychiatric disorders involve sort of a social withdrawal or social impact. And so it brings up lots of interesting questions about how we tip over from sort of acute adaptive immune response to pathological. And again, this is really sort of a bidirectional relationship that we have between sickness and the social brain. So again, to take the current example, we know that infection can lead to changes in social behavior, withdrawal or social interaction, which is adaptive for both the individual and for the sort of kinship group. But social isolation can profoundly impair your immune system. And at the same time, social buffering can enhance your immune system. So it's sort of an interconnected loop. And a few years ago now, my lab showed that microglia, coming back to this cell type we're interested in, not only prune whole synapses within the developing brain, but they actually can very selectively prune specific receptors, including dopamine receptors, which they prune selectively during nucleus accumbens development, in which D1 in particular peaks and then declines again. And if you interrupt that developmental pruning by microglia, you can also interrupt normal social behavior development. So coming back to that initial question, why? Why do we have so many immune cells in our brain doing things like pruning synapses and impacting synaptic function? And I think one major reason is so that they can rapidly impact neural activity in a way that's adaptive for our survival. This also then, of course, brings us to the question of how do we tip from health to pathology, which brings up, once again, lots of interesting questions. In fact, we know that immune activation during specific periods of development, particularly early in life, can increase the risk of a whole slew of different neurological and neuropsychiatric disorders. And this includes neurodevelopmental disorders such as autism, which we're particularly interested in, but even disorders that happen at the other end of the lifespan. So sort of broadly, this is what we look at in my lab. So one fun fact about microglia is they don't come from the neural ectoderms, similar to other cell types. They actually come into the developing embryo from the fetal yolk sac as these sort of primitive stem cells. And they come in very early and they start to divide. And that seeding population of cells is what gives rise to all of the microglia for the rest of the lifespan. So you have this sort of window of when they're coming in, they're dividing, they're becoming mature cells. And that brings up lots of interesting questions about what happens if you have an inflammatory event, for instance, which we model with a broad sort of array of different challenges. During this critical window of microglial development for long-term outcomes. And some of you may notice I have a couple of different trajectories here, and I have that quite intentionally, and that is because we're very interested in sex differences in the way microglia develop, because we're very interested in the fact that there's a sex bias in most of these disorders as well. And in fact, we've shown that there are sex differences in the way microglia mature and the way they react to these various inflammatory stimuli. So we think that there's something quite interesting there about looking at this from that perspective. So as I mentioned, we're really interested in autism in my lab, and so I want to just touch on a study that we've been working on for several years now to try to model some of the social deficits that might be happening in autism. So if you're not so familiar with this disorder. It is, of course, extremely heterogeneous, but at its core, it's really defined by behavioral dysfunction, social dysfunction, repetitive behaviors, etc. And as I just mentioned, of interest for my lab as well, it's one of the most sex-biased neurodevelopmental disorders, or neurological disorders broadly defined as well, so more common in males. And in recent years, there's been a lot of interest in trying to model this idea that inflammation during critical developmental windows might increase the risk of something like autism. And so using mostly mouse models, the idea is that you give something like an infection or infectious mimic to mom, which then induces a maternal immune response, which then impacts the fetal brain in a way that increases risk of pathology. And this has been shown in many mouse models at this point. And the vast majority of them focus on infectious or microbial stimuli, like a viral mimic, for instance. And this is well-founded because there is a literature linking infection to something like autism risk. And it's part of this sort of growing recognition that even though autism is definitely genetic in origin in many cases, there are a lot of genes that have now been associated with autism, it cannot account for the full sort of increase, especially in the rate of autism that we've been seeing in recent decades. So there's growing interest in what are these environmental factors. And I will argue now that infection is maybe not the most relevant thing to be looking at when we think about autism. And again, this sort of goes back to this increasing rate of autism prevalence, which has really gone up and up and up over recent decades. Another thing that has gone up in parallel with this is autoimmune disorders, sort of broadly defined. Almost every autoimmune disorder we have has gone up with the same prevalence. This is quite interesting from an immunological perspective because we know the immune system doesn't just respond to self versus non-self. It actually responds more accurately to danger versus non-danger. And there are a lot of sort of endogenous and exogenous molecules that can activate receptors of the immune system and stimulate an inflammatory response in a way very similar to infection and have sort of a downstream inflammatory cascade. So the work I'll tell you about today is, again, what we've been working on for several years now, which is looking at what we think may be one of the most relevant and epidemiologically one of the strongest links to autism is air pollution. So air pollution exposure during the critical windows of early development has been linked in several very large epidemiological studies now. Now that said, if you look at any single environmental factor and say, what is the link between this and something like autism, it's pretty weak, honestly. Odds ratios are, not so convincing, which tells us that there must be factors that are interacting. And one of the factors that we're very interested in is the potential role of maternal stress. So specifically, whether these two things synergize to increase risk. And why are we interested in this? Well, this is, again, well-founded in the epidemiology literature, in which maternal stress has been linked to an increased risk of an adverse outcome to the same level of environmental pollutant exposure across a whole sort of category of exposures like lead or other air pollutants, et cetera. And this is, for instance, the case with asthma. So it's a pretty striking epidemiological literature, and really the mechanisms are completely unknown. This is also relevant, I think, because of the fact that we know that environmental toxicants and exposures that we experience, we know are disproportionately affecting those with the fewest resources. So modeling something like stress, a reduction in resource, we think probably has a sort of a more than additive, an actual synergistic effect with these exposures. All right, well, we work with mice. So how on earth are we going to model these things in mice? So I won't argue that we can model something as complex as autism or environmental justice in a mouse, but I will argue that we can model sort of core attributes of behavior And in particular, social behavior, I would argue, is just as important for a mouse as it is for a person. They need to find a mate. They need to be able to, have offspring and care for those offspring effectively. And the resources that they have to do so directly impact their ability to do that. So this is the model that we use. Again, in a mouse model, we collaborate with the Environmental Protection Agency to basically have a proxy for air pollutant exposure, one of the primary toxic components of air pollution that we all breathe, diesel exhaust particles. And these are environmentally relevant concentrations, actually relevant for Durham County, FYI. And so depending on your proximity to a roadway and where you live. And we expose pregnant moms throughout pregnancy sort of intermittently to these particles. And what this does is it induces sort of a persistent low-grade inflammatory response in the lungs, including macrophages and neutrophils coming in. And then, as I said, we know it's not just this exposure alone. And what I can tell you is we've done that in mice. And if we look at the offspring, that exposure alone, even though it sounds sort of intense, has no effect on the offspring behavior. They seem completely fine. But what we then do is we take half of the dams in each group and we further expose them to a stressor paradigm. And this is a nest restriction paradigm where we simply take away the bedding and some of the nest material when they're quite literally in nesting mode and building a nest for their coming pups. We take away some of that material. And this has been demonstrated by the lab that developed this paradigm to have a very profound impact on the mom's stress reactivity. So stress hormones are quite elevated. And we see the same thing in our model. So in this case, the 2-hit of diesel particles in maternal stress, you see much higher levels of corticosterone as a proxy for stress in the latter part of gestation. Now, what's really critical about our model is that we actually do the stressor prenatally. So the pups aren't born yet. Before they're born, we give back completely normal resource. So they put back into normal cages. And this is important because if you do this manipulation postnatally, you see big impacts on maternal care. But we do not. We actually see very good maternal care by all of the moms. And so it allows us to sort of simplify our interpretation as much as possible in terms of what the mechanism might be. What we also see in addition to a stress response is I mentioned briefly before that people are interested in modeling the so-called maternal immune activation as a consequence of something like an infection. We actually see almost an identical inflammatory cytokine response to these exposures as you do to something like a viral infection, which is remarkable. The same cytokines go up, which tells us that that sort of initial hypothesis that you have convergence onto these innate immune receptors that drive cytokine expression are actually responding very broadly, not just to something like infection, but to these other factors. Okay, so then what I'll show you briefly is that we see pretty striking changes in the behavior of the offspring as a consequence of this double hit. And again, we don't see any changes in behavior with just one of these exposures alone. So starting with sort of a proxy for communication, what we measure are ultrasonic vocalizations in the mice when they're still babies. So on postnatal day 8, we just very briefly separate them from mom, and then we can measure their vocalizations. And what we see is that both males and females are exhibiting many more calls, so they're calling a lot more. And then we wanted to measure the structure of the call to try to get a sense of, are they simply calling more or are they calling differently? And here we use a machine learning approach to basically classify syllables. And here we found, again, a pretty striking change in the structure of the call. So those pups exposed to the double stressors are actually making much shorter, choppier calls, so less complex calls compared to controls. And then we wanted to see, does this persist into adulthood? Is this just a transient developmental thing? So we can do this in male mice at least, because males will call if you put them in with a female mouse, it's an estrus, they basically start making little love songs. And we can again characterize these calls. And we see pretty much the same phenotype. So once again, the 2-hit exposed males now have much less complex calls. So they're making shorter calls with lower complexity. And this is interesting because we know from a large literature now on ultrasonic vocalizations that males that make these sort of less complex calls tend to be less competitive in terms of mating. And then sort of moving forward to the juvenile period in these mice to characterize their social behavior, which again, we're especially interested in. This is sort of the classic 3-chamber test for sociability in a mouse, in which you simply ask, do they prefer to hang out with another mouse or do they prefer an object? And here we started to see a pretty striking sex difference emerge, because only our males exposed to the 2 hits prenatally have no preference for the social stimulus, whereas females have a normal preference regardless. And for the rest of my talk, this sex difference will be a common theme. And remember, autism is more common in males, so we find this quite interesting. So we've done a bunch of other behavior in these offspring, but in the interest of time, I'm going to go on to what we think mechanistically might be happening in these animals. So as I've alluded to already, we know based on a lot of studies at this point that there's evidence for immune abnormalities sort of broadly defined in at least a significant subset of patients with autism. So not all patients, but certainly some. And I won't go through this in detail, but this is pretty broad, meaning that it seems to affect the innate immune system, the adaptive immune system. You see more things like allergies and autoimmune reactions in families of patients with autism. But what really caught my attention for this was emerging evidence that there may be changes in glia, both microglia and astrocytes, but mostly microglia. And this is based on sort of transcriptomic studies where you just compare, so not looking at the genetic mutations that might be conferred from parent to child, but rather compare a typically developing brain to autism postmortem tissue and ask what are the sort of families of genes that are changing in this case. And again, we see sort of 2 things. Those genes that are important for synaptic function and those genes that are important for immune function. And within those immune families, a specific convergence onto microglial genes. So specifically what you see is a downregulation of genes important for synapse function and an upregulation of genes important for immune function. And notably, as I mentioned, there are a lot of GWAS studies that link, sort of the genetic mutations that are associated with autism. These synaptic genes that you see occurring at a transcriptomic level converge really well with those GWAS studies, suggesting that this is actually the heritable component of autism that we know exists. But those genes that are occurring in the immune system do not converge well at all with GWAS, suggesting that these are de novo or spontaneous mutations and may account for that environmental impact that we're interested in. So the first question we had, are there synaptic changes in our model? Then we asked, where the heck do we look? Because autism is extremely heterogeneous, and we know that there are so many different brain regions that have been implicated in autism. So we started a collaboration with Prof. Sarasa here. And as many of you know, I'm sure, he has these really elegant methods of recording simultaneously from multiple different brain regions as animals are sort of moving around and behaving normally. So in this case, we recorded LFPs from multiple brain regions that have been implicated in autism and in social behavior broadly previously. And this is while they're either interacting with another mouse or with an object. And the question here is really, can you identify what a normal social circuit looks like? And is it altered in our mice? And they first did this in just normal mice. And that allowed them to identify what is essentially a normal social circuit. What are the brain regions that are important? How do they talk to each other? And using this technique, the critical question here is, can you look at this network activity that they've identified and have it reliably predict the mouse's behavior? Because otherwise it's not a very meaningful measure. And so if you look across all of our mice and simply ask that question, do you see more network activity when the animal is interacting with a social stimulus compared to an object? The answer is yes. So this sort of initial identification of the circuit that they identified in control mice is mapping on really well to our experimental mice. And more convincing is the fact that if you now plot the activity of this social circuit relative to the social preference of each individual mouse, you see a pretty strong correlation, positive correlation. Now, this is the case in every group of mice except for one, and that is our two-hit males. So males exposed to the prenatal stressors. So you actually see no correlation whatsoever between the activation of this circuit and their social preference. So clearly they're just not encoding that social experience the same way. And females from both conditions look like control males. So the question for us then was, okay, we're interested in whether we can identify synaptic changes. Where do we look? So I asked Koff, where do we look? Which part is most broken, basically? And then he laughed at me because, it's a circuit. So it doesn't work that way. And in fact, we were very interested in this question, and reviewers were interested in this question. And if you basically plot social preference as area under the curve, so preference for the social stimulus over the object, and then you ask which aspects, which features of the model, whether that be the activity of a single brain region, which is plotted here, or the coherence between individual brain regions, can any one of those alone predict social preference? And the answer is no. So only the circuit activity as a whole can accurately predict social preference, which we thought was pretty striking. But that said, again, we said, but Koff, we can't look for synaptic changes in the whole brain, so where do we look? And there are two brain regions that have sort of the most disrupted coherence, and that is links between the anterior cingulate cortex and the thalamus, and the nucleus accumbens and the thalamus. So those are the two brain regions we're currently focusing on in the lab, and I'll tell you a bit about the work that we're doing on the ACC. We were immediately interested in this brain region because, as you probably know, it's a critical region for social cognition, communication, many of the things that are disrupted in autism. And there have been lots of reports in autism that this brain region is affected, including that it's smaller and that it's hypoactivated. And specifically, you see in a number of studies reduced thalamocortical connectivity. And we can measure these synapses that come from the thalamus. So early in development, the thalamus sends these long-range projections into the synaptic layers of the ACC, which we can measure with a marker called vGlut2. So this is where we started. And so this is all work I should have been mentioning all along by Karina Block, a recent graduate student in the lab. This is all of her PhD work in collaboration with Çağla Roglu here. And so this is a pretty simple question, which is basically counting synapse number. And so we simply take the presynaptic and postsynaptic marker and see where they co-localize to make sure it's a functional synapse. And Karina looked at a number of different time points. At postnatal day 8, based on some preliminary data she collected, that this is when these synapses really start to form. Postnatal day 15, which is when they level off, and then P100 just for a persistent time point. And what she found is that at postnatal day 8, this is just showing males right now, our two-hit males, prenatally exposed males, have more than double the number of these vGlut2 positive synapses compared to controls. But by P15, this reverses. So they now have fewer, and this persists into P100. This is only in males. We don't see this in females. So females have maybe a slight blip at P15, but they recover by P100, and they certainly don't have that overshoot at P8. So now the question becomes, finally, I will talk about microglia again. Now that we know that synapse number is dramatically changed, the obvious question for us at least was, given that microglia are identified in other brain regions for playing a critical role in synaptic pruning, might they be either under or over pruning in this situation? So we can look at this by doing essentially 3D reconstructions of the cells through a Z-stack on a confocal microscope, and then just measure that synaptic marker as well as the lysosomal marker, which is essentially the way the cell degrades the marker inside the cell. And first, Karina, this had not been identified just in normal mice yet for this brain region. So the first thing Karina did was just look in normal development, do microglia even refine these synapses at all? And the answer is yes. So between P8 and P10, hopefully you can appreciate this, at P8, they're just chock full of VGLUT2. So this very, very precise elimination of this synaptic marker between these two time points. So the question, of course, is it altered in our two-hit mice? And the simple answer is yes, but it's actually sort of a complicated variation on that answer, which I'll go over. So we can measure microglial histology with using a number of different markers. One of these is called P2YR12, and one is called IBA1. And we use these markers because they do different things. They label sort of different parts of the cell, which enables us to do these really accurate 3D reconstructions. Usually we use both of these just so we can fully see the cell, and they label both cells evenly. But when Karina did this in our two-hit males, or in our experimental mice more broadly rather, she noticed a really weird pattern, which was that essentially there are patches of microglia that are only labeling for one or the other of these markers, which is really odd. Now, in some ways, we know that microglia are heterogeneous. And in fact, recent single cell databases have shown that early in development, you actually have all these different subtypes of microglia, and they have very heterogeneous function. So maybe that's what we're seeing. Basically, are we seeing just more heterogeneous expression of these different molecules in microglia? And so that's exactly what Karina looked at. She just simply asked how many of these cells are just exclusively labeling for one marker or the other, and found, in fact, that our two-hit males have many more of these heterogeneous microglia. And it's not because of a change in cell number. So they have the same number of microglia regardless of group. So now the obvious question is, who cares? Is there a functional difference in these different flavors of microglia, basically? So again, Karina looked at engulfment of the synapse and phagocytic function in each type of microglia, labeling for one marker or the other or both. And again, found a pretty striking difference where one subtype of microglia are just not engulfing as much. And as a consequence, they are engulfing dramatically fewer synapses. And it turns out that this subtype is over-enriched in our two-hit males. So she's basically identified this very precise subset of microglia that aren't properly pruning synapses, which we believe is then contributing to that overshoot in synapse number at postnatal day 8. So this is sort of the accumulation of what I just told you. So coming back to sort of our model, what I've shown you so far is that what Karina has found is that you have these two sort of very disparate environmental factors, which converge onto microglia and induce this sort of weird heterogeneity where you have a subtype that aren't properly pruning aren't doing their job. And that this leads to this striking change in synapse number, altered neural development, circuit activity, and ultimately behavior. Of course, so far, everything I've shown you is actually correlative. So are the changes in microglia functionally or causally implicated in the social deficit? So to look at this, one final experiment that Karina performed is basically trying to induce the same functional impairment in microglia using a pharmacological approach. So can you basically take a normal mouse and impair phagocytic function by microglia precisely when they should be pruning synapses? And do you then elicit a social deficit in those mice? So that's what she did. She used this thing called neutrophil inhibitory factor, which is actually produced by a hookworm. It actually binds very precisely to the receptor and shuts it down so that it doesn't get ejected by the immune system. And so we can actually put that into the brain precisely during the critical window when they're pruning synapses and have them eat fewer synapses. And that's exactly what she found. So now the microglia that have been treated with NIF are eating about half the number of these glutamatergic synapses, and it completely abolishes sociability at P30. So we can essentially phenocopy or recapitulate the social deficit just by interfering with normal microglial function. Okay, so coming back to sort of big picture here, I've shown you that this sort of convergence onto microglial function is inducing this change in phagocytic function, which is under pruning a synapse, which is leading to sort of an altered connectivity, leading to altered social behavior. The obvious question for us now, of course, is what the heck do we do about it? So is there any way that we can rescue microglial function, especially rescue microglial function if this is happening in people? And so the last just few minutes here, this is a short part, I promise. I want to tell you about a bit of a shift, but something that we've become very interested in the lab, which is how to potentially harness the gut to access the brain. So as you may know, gut dysfunction is actually comorbid with autism and with many neurodevelopmental disorders. In fact, the Center for Autism, where I had an appointment in Boston, upwards of 70% of patients would complain of GI dysfunction or distress to some extent. So this is a huge, huge issue in this patient population. And there's a lot of animal literature looking at this as well. So for instance, we know that mice are so-called germ-free, have no microbiome, have very striking social deficits, and you can rescue it if you give a normal microbiome back. And then clinical trials of fecal transplants in autism patients in recent years have been astoundingly effective. I mean, upwards of 2-3 years later, you still see rescue social function in these patients. So it's a pretty exciting area sort of overall. And another reason we're really interested in this question is that we know that microglial development as immune cells actually depend on a normal microbiome. And so some seminal studies a few years ago now showed that germ-free mice, again, without a normal microbiome, had these really weird microglia that looked essentially functionally immature. And that, again, this seems to particularly affect males. And some more recent work by a postdoc in the lab, Caroline Smith, has shown that if you reconstruct microglia from our two conditions, they look strikingly like germ-free microglia. So they're probably not the same because our mice, of course, have a microbiome, but it led us to ask the question, is there an altered microbiome in our mice? So the first thing that we did was just a very straightforward, did 16S sequencing of the gut microbiome to see if there are changes. And indeed, we found that there are. So, sorry, this is a little bit small, but I'll just explain it. Basically, males again, but not females, show striking changes in several measures of diversity within the composition of the gut, both alpha diversity measures as well as beta diversity measures, which can sort of broadly be conceptualized as how diverse is the composition within a given individual and how might that compare across groups. And then beta diversity being how diverse is the composition between individuals and how might that compare. So you're sort of looking at, do these groups sort of cluster apart such that they're distinct or not? And you can see that very clearly in males as a consequence of these prenatal exposures, they just have a very different gut microbiome. And we can then drill down into the distinct species that you see, or the taxa that you see more correctly. So this is at the genus level. We actually see the depletion of entire genre of bacteria in our two hit males compared to controls. And these two in particular, Bacteroides and Parabacteroides, have also been reported to be depleted in autism. But the question, of course, is, so what? Again, is this actually causal in any way to what we're seeing? So we did the simplest first experiment to go after this question, which was simply to switch the microbiome at birth by doing a cross-fostering experiment. So you take the control pups and give them to a prenatal exposed and vice versa, and you see what happens, right? And so the question is, can we essentially rescue the microbiome changes that we see? Can we rescue the social deficits? And the answer is yes. So when we cross-fostered our diesel stress-exposed pups to a control mom at birth, we rescued both the measures of alpha diversity and beta diversity. This is really hard to see, but they were way out here in terms of the diesel exposed pups, and now they're clustering together with controls after cross-fostering. And we can rescue social behavior using this. So this is all pups that were born to a prenatally exposed dam, and then they were either cross-fostered to either a control dam or another prenatally exposed mom. And when they were fostered to a control mom, they now have normal social behavior. And importantly, it's not because of maternal care. Again, we always measure maternal care. Our prenatally exposed moms, despite all that they've been through, are great moms. They're caring for their pups really well. So that's not what's happening here. So this led to the working hypothesis that, we know that babies acquire their first microbiome through vertical transmission from mom through the birth canal, right? So in the non-cross-fostered condition, we hypothesized that basically these exposures in utero to mom are being vertically transmitted to the offspring, such that they're acquiring this altered microbiome, which is leading to changes in brain development and social dysfunction. But we were wrong. So it turns out, and we've now looked at this every which way, we have thoroughly characterized the microbiome of mom, including her gut, her vaginal microbiome, her milk microbiome. I have a heroic postdoc in the lab, Danielle, that learned how to milk mice. And which is not easy. And we've looked at it every which way. There's no changes to mom's microbiome. So we had to come back to this question, okay, so what's happening to the pups and why do they have an altered microbiome? And of course, the gut is beyond, it's more complicated than simply what's in it. It's not just the composition of the gut, but the gut structure itself. So another scientist in the lab, she was in my lab at Harvard and now has her own lab there, did a really comprehensive assessment of the structure, including the villi and the length of the villi and how intact they are, and essentially the tight junction proteins between them. So lots of things that you can look at. And what she found, again, very striking, hopefully you can appreciate this, is that our prenatally exposed, especially males again, to diesel stress have much longer villi. And this is a phenotype that you actually see often in nutritional deficiencies. So something is definitely going on with the gut of these animals, which we think is actually then conferring the altered microbiota colonization in the animals. So this is my last summary slide here. The revised working model is that we have these prenatal exposures to mom. Because of the pro-inflammatory environment that I mentioned at the beginning of my talk, we know it's inducing a maternal immune response. We believe this is actually leading to altered gut structural development, just as it's leading to altered brain development. The altered gut is then conferring a differential colonization of the gut by the microbes. And this is independent of what's happening in the brain. So you have sort of parallel processes, inflammation leading to altered gut, altered brain development. But we're excited about this because even if the gut microbiota is not causing brain changes, we think it's this potential point of intervention. And we're especially interested in the fact that you can still rescue function by cross-fostering. And we think that probably there's something in the milk that might be really critical. And so we're trying to understand what might be missing in the milk of these prenatally exposed dams, which then confers vulnerability. So with that, I will stop and thank everyone for their attention. Of course, thank my collaborators, Kof and Chala in particular, Karina, who did most of the work I've shown you. The microbiome work is all Caroline Smith and Danielle Rendina. The work was supported by NAEHS and the Robert and Donna Landreth Family Foundation, and everyone in my lab who is fabulous. So thank you very much.
Lovely talk. Thank you so much. Questions from trainees first.
Okay, so I don't know too much about inflammation, but I feel like the common wisdom is that like our environments are full of inflammatory agents, whether it's like flavor retardants or even just like foods that you're not supposed to eat. So I was wondering if there was something specific about the immune response to that. Or if we're all just activating our immune systems constantly, whether or not we're breathing in brain cells.
Yeah, good question. So I get that question a lot. And I always have to make the disclaimer that I am not a toxicologist. So I'm sure that there are differences in these various environmental factors, and the toxicology group here at Duke is actually all over that. Heather Stapleton, for instance, looks at flame retardants and their effects on the immune system. We chose it because of, well, the literature linking diesel exposure, particularly to autism, as well as the literature linking it to altered lung development, because we know that lung development, so for instance, in preterm infants, the two issues you tend to see are lung problems and brain problems. And so there's a lot of interest in communication between those two things. But it was a model that allowed us to essentially impact lung function directly in mom, which I think is a sort of an appealing model. But your question is a good one, and I'm sure that we are having responses. In fact, I think our data, points to that directly, that you can have very different factors in the environment which essentially look dangerous to the immune system. Therefore, the immune system responds in such a way that you have the downstream consequence. So what we think is really sort of hopeful, though, about this research is that there are clearly psychosocial factors that modify that response. And that's where we think maybe an intervention could also be quite effective.
So, right at the beginning of the talk, you were saying that lung function of the immune response is cause temporary social reform, but then it's sort of nice, it's response So I was wondering if these treat the same sort of response as causing temporary changes in adult or permanent changes in the developing infant, and if so, why that's happening, why it's temporary in one case and the other.
So, yeah, that's the million-dollar question for the field. I mean, I think for development, it's in some ways an easier question because, as I've shown, if you hit microglia, for instance, or potentially other immune cells that impact the way the brain develops, if you hit them at a time when the brain is critically developing, you could see why you have this very sustained effect. The question for why that might then also happen in adults where, you know, I mean, the most obvious thing right now is long COVID, right? Why do some people have long persistent symptoms and others don't? And I think that gets right to the heart of the question of how little we understand inflammatory processing within the brain. But there are known risk factors for that sort of thing. So for instance, patients that come in to get treatment for certain cancers and they get cytokine therapies for those cancers, the ones that go on to develop profound depression tend to come in with a low level of inflammation from the beginning. For instance, high CRP or high interleukin 6. Now, why they have that low level of chronic inflammation, that could be quite varied. But so we know a little bit about the sort of risk factors. But it's clear that there's sort of this tipping point between acute inflammation and chronic inflammation. And when the chronic inflammation doesn't get resolved, it's not good.
You're saying low level compared to nothing, not compared to who are at risk for.
Yeah, so people are at risk for depression if they go onto cytokine therapy. Yeah, so there's a threshold.
Yeah, you're saying that their low is still elevated.
Yes, their low is still elevated, exactly. Yeah.
Can I ask a question? So I was interested in the choice of the evolutionary strategy for manipulating inflammation in this model and the MS model and other, like, so can you talk a little bit about whether those hook-borne products are pro-inflammatory, anti-inflammatory, what the cases are, which we expect to help us for this?
Yeah, so that's interesting. So yeah, so this is neutrophil inhibitory factor is just a factor produced by hookworms, which if you can imagine, it's in the gut, for instance, which is where hookworms are, right? They don't want to get ejected by the immune system, so they basically produce this factor which binds to macrophages and essentially silences them for a while, and then they don't get ejected by the immune system. And so we just took advantage of that and used that same factor, put it into the brain and sort of quieted microglia for that critical period. But in the gut, as you know, there are actually a lot of really cool research on how so-called, I don't want to call them commensal, but certainly not quite parasitic worms can do very similar things in the gut where they produce these anti-inflammatory or occluding sort of molecules so that you dampen inflammation, and then that has beneficial effects. And it's effective in MS, it's effective in IBS, Crohn's, like a whole slew of different autoimmune sorts of disorders. Should I look at this?
If you can hear it, you just want to unmute.
Oh, me? Yes? Can you hear me, Staci?
I can hear you.
Okay. I
But
don't
you
know
didn't
if I'm
actually-
cutting them.
She didn't actually have a question, though, did she?
No, I do have a question. I do.
Oh, okay. Okay.
Thanks. I thought that talk was awesome. I thought it was an especially awesome example of how you can work in a subfield with so much jargon and make such an accessible talk to people who have such a different background. That was so beautiful. Thank you. My question is, so I'm going to be like CNN, or whoever you most don't want to cover your work. And so if I'm CNN, right, I say, okay, what I took from your talk is that if you want to decrease your chance of ASD as a prospective mother, you should go move somewhere with no pollution of any kind. You should get a massage at the spa every day so you have no stress. And you should, make changes such that the postnatal environment is compatible, whether it's through nutrition, milk, whatever, with having the most robust microbiome. What out of what I just said strikes you as like the most wrong possible interpretation, or the thing we're the furthest from actually being able to make claims about?
Yeah, I mean, I do get this question often, and they're all incredibly valid points. I think part of the goal of what we do is to try to understand things from a mechanistic perspective, to actually inform policy in a way that you can say, look, this is no longer correlational, because of course you can't go beyond that in most human studies. This is not just a correlation. We show it mechanistically. This is real. We need to change policy to, to benefit health, right, in a way that has more support and environmental protections and the whole bit, which is a high bar, I know, but so I guess from my perspective, that's what we do in terms of why we do the research. Now, when we do the mouse model, we're not even modeling the worst of it, because as soon as they have their pups, they're back in their, comfy cages. There's no pollution. There's nothing happening. So of course, in human populations, that's usually not the case. One thing I've been actually really interested to know, this is probably not really answering your question, but I'm very curious to know what the effect of mask wearing during this phase might actually have on diseases that are associated with air pollution. As much as we all are annoyed by them, especially when you're walking around outside, but there's indoor pollution as well. So it's possible that there are very simple mitigation strategies from that perspective. But what I will say again, though, is that again and again, what our research shows is that the toxicant level itself, while it's bad, of course, we want to reduce that, it doesn't seem to be the tipping point. And that's what we don't yet understand why. But the tipping point seems to be the stress exposure to the mom. And so, is that a more tractable place to intervene? I don't know. I mean, those are both hard problems, right? But it's a great question, Elika. It's something we think about a lot. Yeah.
Thanks, Staci. I do think massages every day for all pregnant women.
I think so too.
Right
I totally agree.
Yeah.
Yes.
Thank you.
I think this is a really inspiring point to wrap things up and thank our speaker, and Colleen will be scheduling massages for all of us, I'm sure. Perhaps not, but thank you. And thanks very much, Staci.
Thank you.